Hello, everyone, and welcome. My name is Mac Erno. I'm the chair of the Huntington Society of Canada Board of Directors, and it's a pleasure to be here today. I've been attending these conferences for 12 or 13 years now, and I got to tell you guys, I sure miss seeing all of your smiling faces and connecting with so many of the great people that we've met over the years. But in the format we're in, it's exciting to be here, and in the world we're living in right now, I'm glad we're able to do this. We have, last count I heard, we had over 700 people registered for today's conference, so pretty phenomenal, and certainly reaching across Canada and beyond, so really exciting. I have the pleasure today to introduce our amazing panel from HDBuzz. HDBuzz, as many of you will already know, is the first internet portal for the rapid dissemination of high-quality HD research news for the global community, written in plain language by HD clinicians and scientists. Today, the panel includes 4 distinguished experts. First is Dr. Leora Fox. She is the Assistant Director of Research and Patient Engagement at HDSA and has been working there since August of 2017. She obtained her PhD in 2016 in neurobiology and behavior at Columbia University, where she studied Huntington disease mouse models. She serves as a contributing author and editor of HDBuzz. The next couple fellows are well known to our community. Professor Ed Wild is a professor of neurology at the University College in London, and he's the Associate Director of UCL's Huntington Disease Center and a consultant neurologist at the National Hospital for Neurology and Neurosurgery. Professor Wild has worked on HD since 2005 and leads a team focusing on clinical trials of new HD treatments and studying cerebrospinal fluid to understand HD. Since 2009, he has been collaborating with Dr. Jeff Carroll to make HD research news accessible to the HD community through HDBuzz. Dr. Jeff Carroll is well known to us as well, and he is an assistant professor at the Behavioral Neuroscience Program and the Department of Psychology at Western Washington University. His scientific interest is in the understanding the systemic effects of Huntington's disease, especially metabolic changes, which he studied using a mouse knock-in model of Huntington disease. As well as conducting research, Jeff is a member of an HD family and himself carries the mutation which causes the disease. Dr. Rachel Harding is an HDSA Berman Topper Career Development Fellow working at the Structural Genomics Consortium at the University of Toronto. Dr. Harding researches the 3D structure and shape of the Huntington protein to try and understand how this molecule works and how this Huntington disease mutation might affect this big complex molecule. Thank you to these distinguished panel members for making time for us today and taking the time to be here. Let's get started. And we will kick off, guys. So we'll get everybody to take their cameras and on and off mute. And I think each one of you are going to introduce yourself, so please go ahead.
Hi, everyone. Well, we've been very generously introduced by you, Mac, so I think we can probably skip any further introductions unless there's anything that any of us would like to say. I think Jeff maybe wants to make it clear that he's an associate professor, not an assistant professor. I'm a professor, so I have no corrections to make. But it's really lovely to be here with you guys, albeit not in person, and I'm looking forward to the day when we can all hug each other again. So let's crack on, because there are questions in the chat already, and I know there's 218 people listening. It's clearly not been a great week for HD research news, but research is about the quest for truth, and we want to find out the truth of what drugs work and what drugs don't work. And if a drug doesn't work, we need to know it doesn't work. Or if it works, but was tested in a way that didn't show that it works, well, then we need to figure that out. So I'm sure we'll have lots of opportunity to answer specifics about the Roche program, but perhaps a more upbeat place to start would be a question that came in from Jackie Stock, who says, wondering what you believe are the most promising therapeutics currently being researched. So Huntington's disease is caused by a mutation in the HD gene, which we call HTT. And to my mind, that's still the right place to focus if we're going to come up with treatments or get treatments into humans that will make a real difference to the progression of the disease. And that is, as I've said previously, something that we have in the bank. That piece of information makes it it gives us a unique head start when it comes to developing treatments. And the first Huntington lowering drug, which was put into a phase 3 human trial, hasn't apparently panned out great. Although we need to figure out, we need to hear the full story before we know for sure. But Huntington protein and the Huntington gene still are the top priority targets. So, the first question to my mind is, how else can we lower Huntington protein, and how can we do that in a way that's hopefully going to have a better outcome than the trials that we heard about this week. And so there are 2 other Huntington lowering trials using a similar approach, but they are only targeting the mutant or abnormal copy of the gene, and those are trials from Wave Life Sciences. The first gene therapy trial, which is a permanent alteration to the way the brain handles and produces Huntington, that is already underway, and we've had no safety concerns from any of those Huntington lowering programs. And Novartis is starting its Huntington lowering trial later this year. So 3 Huntington lowering trials are beginning or underway in 2021, which is pretty good. And then just around the corner, there's a number of other Huntington lowering trials using new technologies. But there's also the opportunity to step back and really make the most of data about other approaches that are to do with the HD gene, but aren't necessarily targeting it directly. And I feel like I've spoken enough, but so maybe Rachel wants to talk about, Rachel's the Huntington expert. So Rachel, why don't you talk about some of the other approaches that are to do with the HD gene, but aren't targeting it directly?
Yeah, so there's lots of quite interesting bits and pieces in the pipeline, and I think like a really important message to keep giving to folks is that the HD research community by no means put all their eggs in one basket, and there are lots and lots of different kinds of research being done to try and understand firstly how Huntington's works, but also how we can make new drugs and therapies for patients. So although there is disappointing news from this one trial, there is actually a whole other amount of work being done by lots and lots of different people all around the world, and that's really exciting, especially for a rare disease like Huntington's. So some of the other things that people are looking at include topics which folks who read HDBuzz might have come across already. So one of those is something called somatic instability. And this is a interesting finding that we've known about for a while now, which is that the repeat length, so we all have the CAG repeat length in our Huntington gene, and that sometimes that can change and it can get slightly bigger or slightly smaller. And it seems that the changing in this repeat length is really linked to the way that the disease progresses. And so there are like companies which are interested in trying to understand how that works, and have a clear idea that we know the mechanism and all the steps in the science. And one of those companies is called Triplet Therapeutics, and they currently have underway like a really interesting natural history study to like really look at patients in detail and understand disease progression. And that's also going to be such a wealth of information for the community, because it will help advise people and clinicians like which patients might be best suited for which treatments when, which is another really important part of the puzzle in treating folks with Huntington's disease. Ed, I'm not sure if there was other things that you wanted me to mention at this point.
No, I think that covers it. And to delve into specifics, the main company that's already running a research study to do with this somatic instability, or kind of freezing the size of the CAG and stopping it from growing. Mm-hmm. The main company that's already running an observational trial is Triplet Therapeutics, and they're hoping to launch a clinical trial. We know that in the near future. And then Locus 23 is another company that's trying to do something similar. So, Ed,
could I just say one last thing? And Lindsay Greer asked a question in the chat as well about how the ongoing natural history study for triplet differs from Enroll HD, which is actually a great question, Lindsay. Hi, Lindsay. because there are observational studies, and we're always telling people go into Enroll HD, and that really is the main observational study for HD patients. And kind of 2 points about triplet. One, the reason that we know somatic instability is an important target is from studies like Enroll HD, because the thousands of DNA samples in that study were sufficient to give investigators the ability to look at tiny variations across people's entire genome and all the genes, their whole genome, not just Huntington, and say, are there variations, like naturally occurring variations in their DNA that make them have HD onset earlier or later than you would expect? And in fact, they found all these little variants that all, it would be it's surprising how often those variants occur in genes that have something to do with the process of DNA repair. And it suggests that, bolstering DNA repair, or in fact, even inhibiting parts of it, might help us have earlier or later onset of HD. So the whole reason companies like Triplet and Locus 23 even know that they should go after these targets is because of HD patient samples that were analyzed by investigators in the GEM HD consortium using Enroll HD samples. And to your specific point, Lindsay, about how is the observational study of Triplet different than Enroll HD? Sometimes drug companies, if they have a very specific way that they think their drug works, or some very specific marker that's like highly specialized and different than the general set of things we look at in Enroll HD, and I don't actually know the specifics of what Triplet needs, but there must be some kind of information or data that they want that's not part of the sort of standard menu of things that people look at in Enroll, and that would be my guess. So if you see these small observational studies for companies that are coming in with like a new technology or a new idea, it's probably because they need some specific human baseline data to help them design their experiments better.
So there's an interesting question from Jodie Ann. Jeff, it's addressed to Jeff, but he may not be the best person to answer it, because the question is, Jeff, did you notice any changes from the injections? But I can exclusively reveal that Jeff hasn't been a participant in any clinical trials, or not any Huntington trial anyway, Huntington lowering trial. Jeff has donated his spinal fluid for observational study research, but hasn't been in a drug trial. But
I could be in a trial. You don't know everything about me. I have secrets, but I'm not
very little.
In a trial.
Jeff, you're not an interesting person. I'm sorry. If that's a shock. It's an important point though, because many people who are in clinical trials, and particularly many people who've been in the Roche tominersen trial, have felt that they have been benefiting from those injections. And it's possible that this drug or any other drug in a trial can produce benefits in a certain group of people and still overall fail, or the trial gets halted for some general effect or a specific effect that is seen in some other group of people. And the important thing about the kind of wind down from any trial, whether it finishes on time or early, is to try and figure out whether there was really a group of people in whom the drug was effective, and whether there's a what we call a therapeutic window. In other words, is there a way of giving the drug that produces benefit overall, or where you can predict which people are going to benefit, and then you only give it to those people. And so that's I don't know whether we're going to end up at that point. And the other thing to bear in mind, of course, is that in any trial, especially a trial where there's a lot of hopes pinned to it, there is undeniably the placebo effect, which is that people in a trial generally feel better because they're in a trial and they know that they're standing and fighting and they're contributing to the fight and maybe receiving something that's therapeutic. So and some people who have felt a lot better during this trial will probably discover eventually that they were actually receiving the placebo injections, which sort of underscores that point. It's unfortunately, those of us who've been running in trials for some years are all too familiar with how it can feel like you go off a cliff edge when the trial stops, whether that's because it's halted early or because it reaches its natural conclusion. And so far in HD, we've only ever had negative trials, really, clinical trials. I mean, trials with clinical endpoints. So the main thing I think is to reach out to your trial team and let them know how you're feeling, and if you need support, it's there for you. We can when the data come out, which I think will happen soon, we can figure out if there is a group of people who are benefited overall. And I want to let Leora tackle one question, but there's a very quick question which I hope I can answer factually. Someone asked which trials have been halted. The Generation HD1 trial is the main phase 3 trial, and that's dosing in that trial has been stopped permanently. So no one in Generation HD1 will receive additional doses of study drug or placebo. The Gen Extend study, which is the open label extension that captures lots of patients from different arms of the Roche tominersen program, that dosing has been suspended in that program, which may or may not be an important distinction. But I think, as far as we can tell, that leaves open the possibility that patients in Gen Extend may, at some future date, be invited to go back on treatment with some altered dosing or something else, some other approach to tominersen. Very early days to tell, but those are the two main studies. Nothing beyond the tominersen Roche program is affected. So every other drug company or everyone else who's developing drugs for HD, we all know that the cause of Huntington's disease is the HD gene and the protein, and so it was a good idea when we started the program, and it's still a good idea to target the cause of Huntington's disease as a means of tackling it.
I think it's
Leora,
probably important to say that as well, that any information that we can glean from this trial will probably be helpful to the other trials that are working on Huntington lowering as well. So, this shouldn't really be seen as a failure. It's more that, it's been halted, and there are things that folks are going to learn from this, and there's probably going to be lots of really important information that scientists and clinicians will be able to get, which will hopefully make us more successful in the future. So, yeah.
Switching gears a little bit, I saw a question about having HD-free babies, and Christian asks, gene editing technology to create babies, how is it implemented today to avoid HD? So it is possible for a person who has the HD gene to have an HD-free child with the help of science. But this, at this point, this does not involve gene editing. So we're not taking embryos and editing out the HD gene and putting them back. This process is usually known as PGD Ivf, pre-implantation genetic diagnosis, and in vitro fertilization. So when the embryos are created and grown in a dish, and actually I'm not an expert on what that process involves, but basically you can screen those embryos to determine whether they have the HD gene or not, and simply implant one that does not. And this is possible to do even if the person the parent who is at risk for HD doesn't want to find out their own gene status. So there are ways to do that testing either anonymously or in ways that check for the parental DNA without determining whether the HD gene is there, and just ensuring that whatever embryo is implanted in the uterus is then HD free. But we're not at the point where we can actually do gene editing like CRISPR on an embryo and edit out the HD gene.
And Leora, if I can just hop in here,
go ahead.
As I was going to say, as someone who's done PGD Ivf, obviously my wife did PGD Ivf. I stood around and did absolutely nothing. But having closely observed the process, it's really remarkable. And this week reminds me, I've had people over the last couple of years come up to me after I've talked about PGD before, with my family's experience, and had them say, "Oh, listen, the Roche study's going on. There's going to be effective treatments in a couple of years, so I don't need to do PGD." And this kind of reminds me that, everybody's decision is personal about whether to pursue PGD or not. But today, if I was doing it today, I would still go through PGD Ivf because, until we have effective treatments, PGD Ivf is the most effective thing we have. And we don't need fancy things like CRISPR. When people heard about CRISPR, they thought, oh gosh, now we can edit embryos. Like, we don't need to. We can already select them with PGD Ivf. And I think that somebody in Canada will have to correct me, but I think most provinces in Canada it's even covered now. So really encourage people to keep thinking about PGD.
Right. So having a kid, if you have the HD mutation, is like if you want to guarantee an HD-free kid, you can do it through PGD Ivf, because it's a bit like tossing a coin, and it's, as we know, the genetic situation with HD is heads or tails. So when someone's tossing a coin, you don't need to like have a robot make you a special coin which has a head on both sides. You can just choose which side. With PGD, you're basically choosing heads from the existing coin. Does that analogy work? That's why we don't need gene editing.
I forgot how analogies work. Are they supposed to make it more clear or less clear?
Okay.
I know less than I did, but yeah.
Thank you.
Do PGD.
Appreciate your input.
There's a question in the chat from Stephen Palmer, who asked, does anxiety or stress exacerbate or increase somatic instability? And yeah, this is a good question. So to the best of our knowledge, this is not the case right now. And what we really think this is about is, I think Jeff touched on this a bit earlier, is that there's increasingly more and more links between the way that the DNA in our cells of our body is maintained and looked after by a huge number of systems, which we call DNA damage repair. And it's actually these protein molecules and how they work, which seems to be more important for somatic instability. I actually popped the link for the HDBuzz article that we wrote about this, because it's a little bit to get your head around. But yeah, so to the best of our knowledge, that is not the case. But I mean, I think there will probably be genetic factors which would make it worse or better. And that's something that we're trying to study more at the moment. But it's also important to remember this is somatic instability. So this is very small changes happening in some cells of your body. It's not, your CAG number isn't increasing more and more exponentially all the time. These changes are generally quite small, and then also not inherited in the same way as you would from other CAG expansions we're talking about.
So can I ask a follow-up question, Rachel? So
yeah.
I'm wondering whether what might be motivating the question is that scientists sometimes talk about cellular stress as being something that can kind of damage DNA, or it kind of puts cells into a special mode where they're trying to protect themselves, and that's got something to do, may have something to do with DNA expansion. But when we use that term cellular stress, we're basically talking about a kind of artificial ways of putting cells to the test. So we can like starve them of oxygen, or we can treat them with poisons and stuff. And it's a way of exploring how cells behave if they are unhappy. But it's that kind of cellular stress is not the same as the stress that we experience in our everyday lives, stress or anxiety. And so I think the important thing probably to emphasize is that there isn't anything that you guys need to be doing that we know about that will keep your CAG the same size. At the moment, this CAG somatic instability is something that you won't know about, because it's sort of you won't feel it happening, and it's not something that any of us has any control over. It's kind of a genetic hand that we were dealt at birth. What we want to do is develop drugs that will make a difference in the favorable direction of preventing the CAGs getting bigger.
Yeah, I think this is a complication of scientists trying to use normal words that explain very complicated things that they're doing in experiments in the lab. So yeah, like Ed says, when we're growing cells in a dish, we'll often stress them in different ways by adding like really quite harsh chemicals and other things. And that's part of our experiment, because we're trying to recreate situations that are, like what we have in a human brain, for example. And it can be very difficult to do that when we're taking a system in isolation. But what's really interesting about this is that this is a completely different way of thinking about Huntington's disease. This is not a new discovery, but it's a new way of thinking about treatments. And this is being followed up by so many different groups, both people who are working in preclinical research. So people like me who are at a bench in a lab with our white coats on, trying to get experiments done at all hours of the day, but also people who are thinking about how we're going to treat patients. So the whole way through, as soon as we find these things that we think might be good ways to make drugs, lots and lots of people are working on it. And I think we're going to be hearing more about different types of therapies which target somatic instability in the next year or so.
Cool. So way up in the chat, we had a question about dementia. Irene, I think it was, asked a question about dementia. She just wants us to talk about it a bit, I think, and how it pertains to HD. So the thing about dementia is that it's a bit like stress, in that it's one of these words that has a meaning that most people understand, and then to scientists and doctors, it has a very specific meaning, and there's some overlap, but they may not necessarily mean exactly the same thing. So when people think about dementia, what they're usually talking about is Alzheimer's disease. But actually, when neurologists talk about dementia, it's a description of a syndrome. It's a kind of what we're saying is that we use the word dementia to mean a progressive loss of thinking skills or loss of thinking ability, cognitive ability. And Alzheimer's disease is the commonest cause of dementia globally. But there are many other things that can cause dementia. So, for instance, having a poor blood supply to the brain is a common cause of dementia, or there are other things like frontotemporal disease, which causes dementia. And actually, to neurologists, Huntington's disease is one cause of dementia. It's actually the commonest genetic cause of dementia. Huntington's, although is rare, is actually a commoner cause of dementia than the genetic causes of Alzheimer's disease, for instance. So, but and that might seem weird because you speak to and you look at your loved ones with HD, and they're not forgetful. They know who you are. They remember their lives and their experiences. But what they have is problems around maybe planning and their thinking may be slowed, and their personality may be different from what it was, and so on. But they're clearly very different from people you may know who have Alzheimer's disease, who are basically, the main thing you notice about that is that they're very forgetful and they don't remember their previous experiences. But nonetheless, we call all of that stuff dementia. But the dementia in Huntington's disease has this very specific kind that kind of matches the parts of the brain that are particularly affected by Huntington's disease. So the frontal lobe, this bit here, where I used to have hair, is that's the bit that looks after kind of personality and behavior, and loss of the function of the neurons there is one of the things that causes HD people to be kind of a bit more irritable or to be a bit more impulsive. And then the connections to the deep structures, which we call the striatum, those can contribute to things like the way that HD patients tend to be a bit more like obsessive, or to have a love of routine. And some of the other kind of things to do with low mood in HD. All of that is the sort of we call it frontal subcortical dementia of Huntington's disease. And unfortunately, that stuff is, obviously it's the kind of stuff that can be most difficult to deal with, because it's kind of changing what it is to be a person with HD, or to live with a person with HD. And although, we got some drugs that are pretty good for things like mood and some of the movement problems in HD, we don't have drugs that work well at all for cognitive impairment. However, if we can find something that targets the central cause of HD, which is the HD gene and the protein, whether that's a Huntington lowering drug, or whether that's a genome editing drug, or whether that's a drug that stops the CAG expanding, maybe we would expect that would treat all aspects of the disease, including this thinking problem or cognitive problem.
There's a question here from Lynn Walker that I think Leora wants to cover about does autoimmune disease affect Huntington's in any way?
Well, in fact, I don't actually know the specific answer to that about autoimmune disease, but maybe Dr. Ed can cut in here. But in general, we do know that lots of different other diseases, whether temporary or chronic, can kind of exacerbate HD symptoms in different ways. And that's actually probably a good segue into talking a little bit about COVID vaccination. In general, we've been very encouraging of folks getting vaccinated. I know that at HDSA we were getting lots of questions about whether it's safe for folks in long-term care facilities with HD, or just people who carry the HD gene in general, whether it's safe for them to get the COVID vaccine. And the short answer is yes, absolutely. And we know that it's really important because COVID itself can really be very bad news for HD and HD symptoms, especially someone who's got advanced symptoms. I don't know if anyone wants to add to that.
No, just to say the short answer is yes, and the long answer is yes. There isn't really any kind of follow up to that, except to say perhaps unless you've been specifically told that there's some reason why you can't have a COVID vaccine because you're allergic to it or something else. But you know, the question began about autoimmune conditions, but the general principle is that it's, you know, there are some very rare circumstances in which getting one disease can be a cure for another disease, but those are very, 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 very few and far between. And broadly speaking, if your body is trying to cope with one disease and you get another disease, they'll both make each other worse. And I think that's more or less true for most things with Huntington's disease. If you have Huntington's, it's bad enough. We know that. But if you have another condition to think about or to worry about, then, you know, that can certainly make it worse. However, if your symptoms of HD are bad and you find out along the way that you have another disease, it's quite possible that treating that other disease might actually also relieve some of your HD symptoms if those two diseases were interacting. And I completely agree about COVID. I think it's perfectly reasonable to be cautious about vaccines and any new drug that comes on the market. But you know, the facts are very straightforward that COVID is really dangerous. You know, millions of people have died from COVID. 0 people have died from vaccination. And, you know, COVID has affected the HD community much worse than other communities because people with HD, you know, are much more at risk of conditions that affect the lungs, which is where COVID likes to cause havoc. So, yeah, I would certainly echo your comments about COVID vaccination.
So Lynn Walker had a question about CAG instability. She asks, does CAG increase if HD comes from the father? And it's a great question. And it reminds me that when we talk about like the unstable CAGs that we were talking about before, about this so-called somatic instability, it can remind folks that across generations in families, the CAG size can also change. And I want to set this up by saying that we think that those processes, right, the instability that leads to kids having higher or lower CAG counts than their HD parent, and the somatic instability process that we've been talking about are probably pretty different, and they're controlled by different genes. And the somatic instability that Triplet and Locus and these other companies are going after is happening in the brain, and you don't make babies with your brain. So that's not the kind of instability that's going to lead to, you know, having a kid that has a higher or lower CAG repeat. But that kind of instability across generations does happen. And for reasons that we don't totally understand, it tends to be exacerbated or worsened or more prevalent in males. So large jumps in CAG repeats are more common in males because of the way they make their germ cells. And so it tends to be true that there might be larger jumps if a CAG expanded CAG repeat is inherited from dad. It doesn't necessarily mean that's the case, but it just means it's a risk factor. Ed, did I clinically miss anything there?
Well, I mean, people definitely use their brains to make babies. That's the first thing I need to say. The second thing would be they don't only use the brains.
I've been doing it wrong.
What do you mean by men make germ cells differently? What's a germ cell? People are going to think you mean bacteria, but you don't. You mean sperm and eggs. Let's get biological.
Yeah, and probably this is not absolutely like rock solid biologically, but the deal is that the DNA that's in egg cells is synthesized and made actually very early in the development in females. And sperm cells, on the other hand, are continuously made throughout a male's whole life. And so we think that the DNA repair processes probably during aging in males affect their sperm cells more than those processes are affected in females because their eggs are actually made quite early. So that's probably, although not absolutely sure, why that happens.
That does make sense. Jody Ann has asked, would risperidone use complicate COVID-19 symptoms? So risperidone is a drug that belongs to a family that we call antipsychotics or neuroleptics. And that's because these drugs, which work on the dopamine system, were first developed and are still widely used for treatment of psychiatric conditions like schizophrenia, if you prefer. And so many people with HD end up taking them and benefiting from them because they are often helpful in the right patients for movement, extra movements in HD or chorea. They can be helpful for some of the behavioral symptoms, so they can kind of even out people's mood in many cases. And if people are losing weight, they can also help people to stabilize the weight. So yeah, lots of people take drugs like risperidone, olanzapine, quetiapine, Seroquel. They go by lots of names. Would it complicate COVID-19 symptoms? No, I don't think it would. To be honest, I think the way that, apart from the vaccine, which is the number one way to protect yourself against COVID, I think the best way to, as an HD patient, to protect yourself against COVID is to be medically as fit as possible, which means, exercising safely outdoors, following your physiotherapy regime, checking in with your neurologist and your psychiatrist if you have one, and trying to get your general medical health as good as possible. And if that includes a recommendation to take risperidone or any other neuroleptic drug, well, that's probably going to be helpful for keeping you safe. I don't know of any reason biologically why risperidone per se would alter your story with COVID if you were exposed to it or if you got it. I don't think there's any involvement of the dopamine system biologically in the way that COVID affects the body. The only thing I could think of is if you were taking a dose of risperidone that's too high, you could end up very sort of drowsy and sedated, and that could affect your breathing or your ability to sort of keep your lungs clear. So I think, but again, that would basically come back down to take the dose of COVID, take the dose of risperidone or whatever other medication
Don't do that.
that you've been recommended. Yeah, don't take a dose of COVID.
Thanks, Ed. There's a question from Jennifer Lukes, I think her surname is, about juvenile Huntington's disease and what is the cause of this. So something I think most people here today are familiar with is that Huntington's is caused by the increase in the number of the CAG repeats in your DNA for the Huntington gene, going over a certain number. We also know that in general, people who have longer CAG numbers or higher CAG numbers, will have the disease earlier. And I think juvenile HD is commonly characterized as having 60 or more CAG repeats. So this is really at the much higher end that we see in patients. And if you follow that trend of a longer number of CAG repeats will lead to earlier onset of symptoms for patients, then this is where we start seeing Huntington's in kids. And so that's the root cause of that.
Follow-up question on COVID vaccine from Jill and Jerry. You've implied that one is at greater risk having HD and contracting COVID. Does that affect eligibility for COVID vaccines? There's been, I think the answer there is that there's probably a lot of variability between different countries and different regions. So that would be one to check with your local health provider or local health system. In the UK, the rollout has been very strictly according to age and health risk, and one of the high priority groups were patients with long-term neurological conditions. So what we've been, I'm on the advisory panel to the Association of British Neurologists on Neurogenetic issues. So one of the sort of conclusions, one of our recommendations basically was that people who have so-called manifest HD, in other words, HD with symptoms, they should be considered at higher risk, because even from very early on, you guys, I'm sure you know that, even if someone's doing really well generally, they will often have things like they'll be more likely to cough during meals, or the food goes the wrong way if they're watching a soap opera during a meal. And those sort of subtle things are exactly the sorts of places how COVID finds a way into the body. If there's any kind of swallowing or breathing or sort of saliva issues, those are what puts you at greater risk of COVID. So if there's any kind of health-based eligibility criteria for COVID in your area, I would say, look to the UK if it helps, because in the UK it's very clear that patients with manifest HD very much do qualify for earlier access to COVID vaccination.
I see Tamara Mowry has kindly put in the comments that her mum got the vaccine in Ontario, even though she doesn't qualify in the current age group. So that's really helpful. Thanks so much, Tam, for letting us know about that. Good.
There is a way. Anne has asked, does HD affect your sleep? Anyone want to?
Sure. I'll take it. So absolutely HD affects sleep. And I think, this is one of these places where we actually have really good evidence, both from humans, but also from HD animal models, to suggest that not only is sleep disrupted in HD, but that helping that sleep, like symptomatically helping it, even just with like normal sleeping pills, not special HD sleeping pills, but like whatever treatment your doctor recommends for helping you sleep, can not just make the sleep better, but can actually improve, say, HD symptoms, which can be exacerbated by bad sleep, the cognition, brain fog, that kind of stuff. So I think there's really good biological evidence for sleep disruption being a very early and very consistent part of HD. So melatonin, the natural hormone that our bodies make to put us to sleep, is reduced in HD patients even very early. So I'm not sure, Ed, maybe I should turn to you for medical advice, but I would just say personally, I know in my family, I've been recommending folks take melatonin as long as they tolerate it. And I think it's really, really helping people. And I think in general, I'd say both scientifically and anecdotally, like seeing people with improved sleep, it really seems to improve not just their mood, but their HD symptoms as well.
Right, exactly. I don't know whether there's any. I'm sorry. You go ahead, Leora.
I was just going to add that HTSA is currently funding a study on melatonin. There isn't a lot of direct evidence in HD, but I think it's important to understand that in a more scientific way.
That's great.
Yeah, I agree. I agree completely. I think it's really important whenever we find a feature of HD that might be treatable, to treat it, because of the inconvenience or loss of quality of life that thing is causing. But sleep is such an overarching thing, like it's something that has such big knock-on effects for everything else. So if you're not sleeping, you're going to feel awful, but you're also going to have lower mood, and you're going to have worse anxiety, and your movement control is going to be worse, and your attention is going to be poor, so you're more likely to experience swallowing difficulties. And so basically sleep is one of these things. If we can treat it, we absolutely should. Melatonin is helpful for a lot of people, but there are actually many different ways through which HD can affect sleep. So in some people, they sleep badly because they lie awake at night worrying about stuff. So it's actually nocturnal sort of anxiety that's keeping them awake. And for those people, an anti-anxiety medication can often be helpful. So I think the thing to say about that is that don't assume it's just HD and there's nothing you can do about it. Absolutely speak to your doctor. And it's one of those things that's easy to forget when we're seeing patients regularly for a checkup. So if you're sleeping badly, just say, hey, I'm not sleeping that well. Ask me some questions about that, and your neurologist or whoever will be able to hopefully help you to kind of drill down as to whether it's one thing or whether there's 2, 3, or 4 things that might be helpful for helping you to sleep better. I don't know that there's any evidence that improving sleep will actually slow the progression of the disease in terms of the how quickly it progresses, but it will certainly, if we can help with sleep, it will give you a much better quality of life, and there's very often effective ways to do it.
Ed, to your point about management of specific symptoms, I know the first question that we addressed was about what promising trials are out there. And I think we focused a little bit on kind of the genetic side of things. But there's also a lot of of trials that are focused on particular symptoms, managing particular symptoms, or other aspects of HD biology that could help with certain symptoms. And so there's promising research out there that is focusing on helping people to manage those symptoms as well.
That's an excellent point. And some of those symptom control studies will be smaller scale, because if you've got a clinical readout, you don't necessarily need to enroll hundreds and hundreds and hundreds of people across the world. So those are studies that will probably attract less attention than kind of big international studies or studies of things that sound very sexy. But nonetheless, it's super important for us to study things that give people an improved quality of life, because that's what it's all about, for sure. So I don't know who wants to take this question, but here it is from Mike. Are there any studies crossing over with Alzheimer's and non-pharmaceuticals, e.g. quercetin? I don't know that. Does anyone recognize that molecule? That's a new one on me. And how heavily do these types of supplements weigh in? So there is absolutely, the community of HD researchers is also, to a large extent, the community of researchers into other brain diseases. And for instance, we were one of the first, a condition called spinal muscular atrophy was the first in which these DNA-based ASO drugs were tested. We were the second brain disease in which they were tested, and now they're being tested in a number of brain diseases, including Alzheimer's disease and SMA, motor neuron disease, ALS. So there's an awful lot of overlap, and whenever someone in one disease has a good idea, the first thing that people who work on similar diseases or related diseases do is think, well, how could I do something similar? I don't know if anyone knows quercetin, but Jeff, maybe you want to comment briefly on supplements in general.
Sorry, I had to find the mute. And sorry, I don't know that particular molecule either. But I'll say that, when I first found out about HD in my family, I took a bunch of supplements. I took coenzyme Q10, I took creatine, I took blueberry extract, because those were the kind of ones that everybody was talking about all those years ago. Honestly, they're expensive in the US and I'd say North America. They're really poorly regulated. So the steps that we have to jump through to make clean drug products are incredible, but these off the shelf supplements have very poor quality control. And on the hand, you might say, well, okay, but at least it can't hurt. But actually, there have been some surprising studies of supplements, so things like vitamin D, that were the hit supplement of the week. And then it's they did a large scale in aging women and they had enhanced fatality, more deaths happen in people taking large doses of vitamin, whatever. The point is that there's no such thing as a totally inert molecule. And so for me personally, there's nothing right now that if I look at HD and knowing what I know about HD and looking at all the supplements in the world, there's nothing that I think is worth the risk personally, speaking for myself. And so there's no sort of secret treatment off the shelf, that we have good evidence for. And other people, people can tell you that they think, supplement X or supplement Y is good to try. But just realize it's based on opinion and it's not based on published good science. Mac, are we?
We are at that time. I always feel like these sessions could go on forever. Is there a final question you guys are really wanting to address, or are we?
So maybe we could just wrap up by, sharing a few resources related to the Roche news. I don't know if you've got a specific statement about it, Ed. There's not a lot that we know right now. All the information we have is currently based on the public press releases. And there's more information hopefully to come in the near future. But EHA, HDO, and HDSA all did specific Q&As on these topics. And I am happy to share those links in the chat if that would be helpful. Hopefully I'll have time to to find those and paste them.
Yeah, I would simply add, I've been involved in this Roche program and many other drug development programs, but the Roche program I've been involved in for about 9 years. And I can tell you that the people involved at every level at Roche are gutted, really, really sad about this news. And, I know that there's a lot of frustration because there's so little information. But honestly, the core study team that's been running this trial for 4 years or 3 years heard this news on Monday, right? So when it went public, and the way that the information flows in a situation like this is always going to be frustrating, because there are the information that can be released is basically governed by laws about stock trading and share prices and international finance laws and all that kind of stuff. So anything that could affect the share price has to be released in a very coordinated way. And of course, then when you release the detailed data, you have to make sure that you're releasing a really accurate set of data. So, but everyone I've spoken to at Rush is really enthusiastic about communicating the results of the study as quickly as possible. But at the moment, the team just doesn't know what those results are. The safety committee's made a decision, and the actual results and the data from the study will take a while to release. But there's a very firm commitment to get the information out as quickly as possible and as reliably as possible. There are also laws about how a sponsor is allowed to communicate directly to the patient. So most of the communication has to go through the individual study sites and the doctors and the people who run the studies at the sites. So if you do have questions, it's the sites that will need to answer them for now, but there will be big communications, and we've been given to understand that we can expect a clear, open dissemination of the results of the study. In the meantime, keep an eye on HDBuzz. We're going to be writing a story soon about the other trials that are, non-Roche trials that are coming soon, so that we can all remind ourselves of how much progress we have made, and how, even though we had this setback, we are still way further ahead than we were 3, 4, 5 years ago. And even this setback is something that we can learn from.
Thanks, guys. I think a great note to end on. I think these sessions, they always feel like they could go on for hours, and there'd be lots of things to talk about. So, but unfortunately, I'm the one that has to cut us off. So, I want to thank all 4 of you for your time today. It was special note to Dr. Carroll that it's associate professor, not assistant. I am at best a trained monkey that reads what's put in front of me, so I apologize for that. But it's been a great HDBuzz session and fascinating Q&A. I'll tip my hat to all of you guys today for your support and making time. Even though we're physically distanced, I feel like we're together as much as we can be. As with every HSC national conference, I learned so much. I'm feeling rejuvenated and to keep up the hard work and the fight to control HD. Pardon me. I was also incredibly moved by the compassionate discussion and dedication to find new and effective ways of coping with the biggies in life: how we live, how we die, and how we fill the hours in between. Many of you know me and know how connected I am to HD. And I know it's not always easy, and this week hasn't been easy either, but I have to say that I'm especially filled with hope after a day like today. Connecting with all of you is always a big part of that. So that brings me to being thankful. We certainly want to say a big thank you to our sponsors for making this event possible. It's a gigantic undertaking, and it's because of those sponsors that this event is free of charge. We'll thank all of our attendees for taking the time to join us today. We had a huge number, a great response, and of course, thank you to our speakers and our staff at HSC for putting together this conference. It's been a stellar lineup, and I appreciate everyone's participation. And I hope everyone takes the chance to join for the social tables tonight. So no matter what time zone you're in, they're starting in just under 1 hour. So those start at 6 o'clock Eastern time, and it'll be an opportunity to connect with many familiar faces that normally we would see together. So hopefully you're able to join for that. Don't forget to mingle virtually, and we hope to see you there. Thanks, everyone.
Thank you.